our last final talk of the day is by Dr. Reid's Learning Adaptive Group. And this is Do Rats and Humans Learn Behavioral Skills in the Same Way?
Good afternoon, you guys. Our lab looked at Do Rats and Humans Learn Behavioral Skills in the Same Way. My name is Deja.
I'm Kelly Bossert.
I'm Shealy.
I'm Stilwell.
And I'm Margaret Roach.
Dr. Reid's lab for the past several years has looked at behavioral skills in animals. The last group we just saw was pigeons, and we studied rats. And our class, specifically Learning and Adaptive Behavior, looked to apply principles from human skill learning to animal skill learning. This question, however, is difficult to answer because the way in which humans learn how to do a behavioral skill and rats is a bit different, so it makes the question difficult to answer. Human studies have focused mostly on knowledge of results, which is the role of post-trial informative feedback about performance errors during skill acquisition. Throughout the presentation, we'll refer to knowledge of results as KR for short. KR is widely regarded as the most important variable in human skill acquisition, and human studies tend to ignore the other type of feedback, which is the operant consequences of responding, which may be food or timeout. When cognitive psychologists manipulate KR for humans, the subjects get feedback about how accurate their behavior was. Some studies for humans have decided instead of giving feedback on every trial, which would be 100% KR, to instead limit that feedback to about 2/3 of the time, which equals 67% KR. Rat studies have always provided plenty of operant consequences of responding, but to our knowledge, no KR procedures with rats or pigeons have been published that manipulated informative KR, and that is the purpose of today's study.
A highly cited paper in Cognitive Psychology by Schmidt and Bjork provide an empirical review of a surprising counterintuitive feature found in many types of skill learning in humans. Factors that degrade performance during acquisition phases often enhance performance during subsequent retention phases. They argue that introducing difficulties to the learner can enhance training because they trigger encoding and retrieval processes that then aid in memory, learning, and comprehension. Skill learning studies usually consist of two phases: an acquisition phase in which the skill is acquired in the presence of helpful cues and feedback, and a retention phase in which the retention of the skill is measured in absence of those helpful cues and feedback. In animal studies with rats and pigeons, we typically call this retention phase a autonomy phase because we are interested to the degree in which animals can perform this skill by themselves or without that informative feedback or guiding cues. Wolf and Schmidt studied whether KR works the same way with more complex behavioral skills called generalized motor programs. They found that practice with reduced KR was more beneficial for skill retention, even with these more complex behavioral skills. Specifically, their 67% KR group performed better than their 100% KR group in the retention phase.
So in our study design, we had operant feedback delivered in the form of food pellets or timeout, and KR was delivered in the form of a beep. We did not allow for operant feedback, as shown here in black, and KR feedback, as shown in red, to overlap in time. Operant feedback occurred within 0.2 of a second following response, and KR feedback always began after operant feedback ended. Sequence errors resulted in a full 1 second tone, and correct sequence responses resulted in 4 tone beeps over the course of 2 seconds. Food was provided with a probability of 0.5 in order to degrade the correlation between probability of KR and operant feedback. We had 26 naive female rats divided randomly into 3 experimental groups of 0, 67, and 100% KR. Training happened via auto-shaping procedures done prior to the start of the semester by Dr. Reid. And overall, sessions timed out at 45 minutes or until 120 correct trials had been completed by the rats. The acquisition phase lasted 30 sessions where the left-right sequence was guided by blue and green panel lights. KR was provided only following a 2-press sequence with frequency of feedback occurring relative to the experimental group. The autonomy phase then lasted only 10 sessions, and the blue and green panel lights were replaced with white lights, and all KR was removed. This phase assessed how well each rat could perform the left-right press sequence on its own.
So here you can see all three groups for both the acquisition phase and the autonomy phase. You can see the x-axis is the sessions and the y-axis is the accuracy up to 100%. You can see here that the 67% KR group, as indicated by red, is at a higher percent accuracy than both the 100% and the 0% KR groups. Some research has suggested that the 100% KR group should have the highest accuracy, but here you can see it actually has the lowest accuracy, even lower than the 0% KR group. The 0% KR group acted as our control, as these rats were given no KR and only given operant feedback. For these rats, the only difference between the acquisition session and the autonomy session was that the blue-green lights were replaced with white lights. This graph demonstrates that even without KR, learning still does occur. The most important result from the Wolfenschmidt's human study was that, contrary to expectations, the 67% group outperformed the 100% KR group in the autonomy phase. Our rats showed these same clear effects. However, what was different in our study, that was how the 67% KR group outperformed the 100% KR group in both phases and not just the autonomy phase as previously predicted. Finally, you can see here that the 67% KR group outperformed the 0% KR group. This shows that informative knowledge of results is more effective in increasing accuracy than no knowledge of results at all. Rats always receive operant feedback, but this was the first study, as Margaret said earlier, that the rats were getting KR on top of operant feedback, thus showing positive results in terms of increasing accuracy and autonomy.
Okay, so our hypothesis was supported. As you can see, that the 67% KR group did have greater accuracy in the autonomy phase than the 100% KR group. And what we were seeking to find is that we wanted to know if limited knowledge of results was going to help retention, but we also wanted to know if more knowledge of results was better than none. And our results were supported, as you can see. And the difference between the human studies, in the human studies you did see that they said that factors that degraded retention in the acquisition phase would enhance learning in the autonomy phase, but this didn't happen in our study. As you can see, the 67% KR group did better learning throughout the entire experiment. We think the reason for this was the difference between like Dr. Bob was saying earlier, how the rats learn and how the humans learn. So in the human study, they only had one session and it was only about 30 trials. But in our study, the rats had 30 days of sessions, 30 sessions, and each day they could do over 100 trials. So they had over 3,000 trials, and it shouldn't be surprising that with 3,000 trials you're going to learn a lot better than you would with just 30 trials. For future studies, we want to see whether our rats could perform the behavior autonomously a little bit earlier. So as you can see here, maybe at about 10 to 15 sessions, the 67% KR group is already exceeding the 0 and 100% KR group. So we want to see if we shorten the acquisition phase, if they can perform autonomously with the white lights. And also we wanted to see if there's a way for us to measure autonomy after each acquisition session, so that way we can see the development of acquisition over time. A lot of studies are now looking at what they call desirable difficulties, and Dee talked about it a little bit earlier, but desirable difficulties are desirable because they trigger encoding and retrieval processes that support learning, comprehension, and remembering. So sadly, for students at least, this shows something that our professors should do that we might not like. It tells us that instead of them just giving us quizzes over time that test whether it's like quick learning, like, okay, I taught you yesterday, do you get it today? That we should actually be getting a lot more difficult assignments so that we can retain the information over a longer period of time. Obviously, students don't want to hear that, but I think it would help over time if, because a lot of people are psych majors and we're taking the GRE and stuff like that, and we don't remember half the stuff we learned in intro to psych. So that's just something that we learned during doing this. If you introduce small things that are a little bit more challenging, it'll help you remember it when you need to.
We'd like to say thank you to Dr. Reid. This experiment actually occurred over 2 different semesters for the past year. So thank you to last year's Learning and Adaptive Behavior class and our other classmates in our class this year, as well as our reps.
Questions? You gotta ask.
Tim.
Yeah, how are y'all going about explaining the superior performance of rats both in the 0% and 67% knowledge of results and the acquisition phase over the 100% knowledge of results rats?
So just during the acquisition phase is what you're
Yes.
asking about? So the rats that are in the acquisition phase are relying only, it seems, on the KR for their performance. So I know when that translates over to autonomy then, those procedural memories aren't occurring enough for them to be able to do the pattern. But as far as during acquisition,
I think that, okay, so during acquisition, they're just learning the new tasks. So what our study is saying that producing some type of KR, some type of knowledge of results, some type of informative feedback will help you learn something quicker. So even though the 100% KR group was lesser than the 0 and 67%, we think it's really just because of the, you don't want too much information. You don't want to give them all the answers. So when, in Dr. Reid's lab, we compared it to a child. So if you're trying to teach a child how to walk home from school and you want them to remember the pattern, so you don't want to tell them exactly where to go at every single turn. You want to lay back sometimes and let them decide. So giving them 100% feedback and helping them all the time isn't going to help them learn stuff quicker. That answers your question.
Do you think your results have overlapped with my thesis group's results, better memory for negative images?
I personally don't think so. I can't see how you're saying it though. So negative images, I know that for the rats, instead of it being like an image, it's something that they have to experience, so they get a timeout, and this is kind of like survival for them because it's food. So if they don't do something correctly, then it'll result in them not getting reinforced with food, and that's the operant feedback part of our study. And I would just believe that like you said, with memory, it'll help. They purposely try to remember this left-right pattern so that they are reinforced, and I think it's more so to prevent that negative, which is the timeout.
Conversely, I think that it may overlap some because we're looking at difficulties during learning, and y'all are presenting difficulties in the form of images that are more negative and may be more difficult to process. So it could work in the saying that those different difficulties both enhance retention.
Okay, thank you so much.